So hopefully you're seeing the appropriate screen and I'll get started. And I'll be speaking today on prostate cancer inflammatory markers. Are they black and white? Now, prostate cancer is the second leading cause of cancer deaths in American men. And Black men are 2 to 3 times more likely to die from the disease than their white counterparts. So what exactly causes this disparity or contributes to this major disparity? Now, there are multiple factors that contribute to cancer health disparities in general. This list highlights that inequities in living standards, inequities in access to healthcare, which are both facilitated in part by systemic racism, are major drivers major drivers of the disparities we observe in cancer incidence and ultimately in cancer outcomes. But even so, we observe biological and genetic factors that contribute to these differences. And so I'm going to highlight a few factors that are observed specifically in prostate cancer. So first off, this study reports that Black men are more likely to develop prostate tumors at the front or the anterior side of the prostate, contrary to their white counterparts who are more likely to have tumors at the back of the prostate. And this may affect diagnosis from biopsy sampling, since biopsy needles are typically inserted from the posterior or back side of the prostate. And further, the study also reported that independent of race, tumors that are located at the front of the prostate are described as having a potentially worse disease subtype. Now, there are multiple studies that identify prostate cancer genetic risk variants that are more frequent in men of African ancestry compared to men of European ancestry. And finally, inflammation and genes related to inflammation are reported as overexpressed in tumors from Black men. So how exactly does inflammation contribute to prostate cancer? Now, chronic inflammation may be caused by multiple factors, including infectious agents, for instance, infectious agents may be something like a sexually transmitted infection. Their dietary habits, for instance, consumption of meats that are cooked at very high temperatures where the carcinogen FIP is generated. And so either way, these insults typically initiate an immune response where immune cells, including neutrophils and macrophages, are recruited to the site at an attempt of eliminating whatever the insult is. And this is done in association or with the help of cytokines. Cytokines are secreted by immune cells and also help to recruit other immune cells and may have numerous other functions in this process. And so this process of eliminating foreign insults, viruses, bacteria, etc., generates reactive oxygen species and reactive nitrogen species. And these are toxic to the tissue in some sense, as they may cause injury to prostate tissues. And that generates an environment that's conducive to cancer development. Also, chronic inflammation throughout the process continues to facilitate cancer progression. And so based on previous studies that highlight inflammation-related genes as overexpressed in tumors from Black men, my specific question was looking at whether these 3 factors, neutrophils, macrophages, and cytokines, may be different in terms of their distribution by race, and thereby might be contributing to the more aggressive phenotype we see in Black men. So we begin with cytokines. We looked at 4 different cytokines that had previously been reported as overexpressed in Black men. And now in previous studies, these cytokines are typically measured in blood, or you have tissues from tumors that are grinded up, RNA is extracted, and then thousands of genes are assessed. And so the advantage of this particular approach is that we're able to look directly at the tissues. And so we're able to see precisely where the cytokines are being expressed, which cells are making them, and whether they differ by tissue type. Among the 4 cytokines we assessed, IL-8 was the most highly expressed, and it was the only cytokine that seemed to be actually made by the tumor cells, shown here by the blue arrows, as opposed to immune cells that happen to be swimming through the tumors, and we thought that was significant. So we went a bit further to look into IL-8 in these tissues and quantify the amount of IL-8 in our prostate cancer tissues. What we saw is that there is increased expression of IL-8 in cancer compared to benign or non-cancer tissues. We also saw increased IL-8 in higher grade cases, meaning the worse the cancer, the more IL-8 we measured. Now, when we looked by race, we saw that there was no statistical difference in Black versus white men. But we should note that Black men are more likely to be diagnosed with a higher grade prostate cancer. And so it is likely that in previous studies that measured IL-18 tumors from Black men as higher, there may have been a larger sampling of higher grade cases among those Black men, which contributed to that increased expression that was measured. So next we looked at neutrophils. Neutrophils are the most abundant immune cells in the body, and they're the first on site when there's an infection. And so in normal prostates, these are from organ donors, meaning there's no cancer, they're as normal as we're going to see. There are very few neutrophils, as we would expect. We see some neutrophils in blood vessels, as neutrophils tend to swim around searching for, infections and so forth. And occasionally a few neutrophils may be seen in normal glands. On the other hand, in our prostate cancer tissues, we see far more neutrophils, particularly swimming along the cancer cells, and again in blood vessels. And this was true too when we quantified, we saw that there was significantly more neutrophils in cancer compared to benign. When we looked by race, we saw that both in benign tissues or your non-cancer tissues, as well in cancer tissues, there were significantly more neutrophils in tissues from white men compared to tissues from Black men. Now, one of the most common mutations associated with prostate cancer is PTEN loss. PTEN is a tumor suppressor, and it's lost in about 50% of castrate-resistant prostate cancer. We observed significantly more neutrophils in cases with PTEN loss compared to those that had PTEN intact. And I should note that PTEN loss is actually associated with worse outcomes for prostate cancer. But interestingly, PTEN loss is actually less frequent among Black men. So then we wanted to look at PTEN neutrophil, the number of neutrophils by PTEN status separately by race. And what we saw is that among white men, that increased neutrophils in PTEN loss cases is maintained, but that difference is actually attenuated among Black men. And more interestingly, when we use statistical correction to account for the contribution of PTEN status to the amount of neutrophils we saw between races, this difference was actually attenuated. And so this suggests that the observation that cancer tissues from white men had more neutrophils than tissues from Black men may have been confounded by the fact that white men are more likely to have PTEN mutations. So finally, we assessed macrophages, which are well studied for their role in cancer. We use 3 different markers to assess these. The first is CD68. So CD68 is a pan marker. That means it will detect any macrophage subtype. CD80 will detect M1 subtypes, and these are thought to be pro-inflammatory or anti-cancer. And finally, CD163 detects M2 macrophages, which are thought to be pro-tumorigenic, or meaning they help in cancer progression. Now, we observed far more M2 macrophages in our normal non-cancer prostates compared to M1, which was somewhat of a surprise. But what we did see, which was expected, is that in our prostate cancer tissues, we see significantly more macrophages, including the M1 subtype. Now, when we compare by race, we see that CD68, so our pan marker, all macrophages, seem to be more highly expressed in white tissues or tissues from white men compared to Black men. And this was also true for our M2 marker. But interestingly, and as a reminder, M2, that is the pro-tumorigenic subtype. But interestingly, we saw that the M1 subtype marker had significantly more expression in Black compared to white men in benign tissues. And so there's actually very little known about M1 subtypes function specifically in prostate cancer. And so we do need future studies to assess whether that function, the function of M1 subtype, might be contributing to some of the aggressiveness we see in Black men. So I have to highlight some of the limitations of this study. In the first instance, all of the cases are self-identified race, not measured genetic ancestry. So what we're assessing here are really differences of sociocultural groups, not necessarily identifying genetic associations. Also, immune response and inflammation as they relate to cancer are very complex processes. So there are many other markers that are involved, many other immune cells that I haven't mentioned. And so my study was specifically looking at these three factors. And so just because they're not, well-defined differences, it doesn't mean that other players are not different. For instance, there are a number of studies that have shown increased number of plasma cells in tumors from Black men, and that has been pretty consistent. And also that increased number of plasma cells is reported or thought to be associated with improved immunotherapy response, which is good news for Black men with prostate cancer. And finally, these comparisons occur after the storm, meaning, this study is a snapshot in time after cancer has already ravaged the prostate. So we're unable from this particular study to determine which inflammatory factors may have actually been involved in the initiation stages. And also when things are sort of this far gone, it's kind of hard to say what was there before, what was not, what's really contributing, and whether everything just kind of looks the same because it's already gotten to that threshold of chaos. And so we do observe increased infiltration of innate immune cells in the prostate tumor microenvironment of both Black and white men. And potentially the composition of these immune cells may vary by race, but we do have to consider that differences in grade or mutation frequency and other non-race related factors may be confounders in terms of the differences we do observe. And so the fact is Black men have the worst prostate cancer outcomes. And it is well accepted that inflammation contributes to this aggressiveness, prostate cancer aggressiveness. My specific question was really aimed at determining whether the distribution of three particular factors, cytokines, neutrophils, or macrophages, may differ by race and thereby might be contributors to prostate cancer disparities. And so we do observe differences, but some of them may be confounded by non-race factors. And so it's really important that we have more studies, future studies that can better decipher which markers, if any, contribute to prostate cancer racial disparities. So the bottom line is, it's not that black and white. And I'll happily take questions.
Great, thank you very much, for a really interesting talk, particularly highlighting these really important differences that I think to date have not really been looked at in any type of detail. There's a question from Bonnie McLean. In your opinion, does this influence the role of race as a social construct? Scientists often claim human variation and genetics as demonstrations of race as biologically real. Do you think environment and culture, perhaps Black men are less likely to go to the doctor early in disease course, for example, are the actual drivers?
Yeah, so I would say that there are multiple things happening. So in the first instance, yes, race is a social construct in the sense that, there's not a genetic code that decides that one person is genetically distinct from the other. However, those social constructs come from a very long history. So for example, typically persons of African descent who will have very similar physical features and similar culture and so forth tend to be classified in one group. And so it means they also share a number of genetic commonalities. And so this is why I made a distinction of this being self-identified race as opposed to genetic ancestry. There are ways for us to use genetic phenotyping to identify a group of people, for instance, who have a certain percentage of African ancestry. And there are, in fact, factors that are being teased out that seem to be more common among that group versus any other group. At the same time, it doesn't mean that every difference we see is based on something you inherited because of your genetic background. Absolutely, as I highlighted in one of the earlier slides, environment plays a massive role. And so if you have a country such as the United States, where those social groups experience very different lifestyles, right? They have very different environments, they're exposed to different levels of pollution, they have different access to healthcare. Absolutely, that's going to contribute to disparities. Particularly in prostate cancer, though, we see that even with that, even if we were to make everything equal, we're still seeing some differences. And so it's important here to understand that we're not really trying to sort of say, hey, if you're Black, you're just going to get prostate cancer, you can't do anything about it, it's just inherited. But it's more sort of trying to tease out what unique things might be happening in one subset so that we're better able to diagnose, for example. So when Black men do have the option to go to a doctor, if there's something pretty unique about their genetic makeup, that might predispose them to a certain type of cancer, we know how to treat them. Or if we find factors, for example, a particular immune cell that seems to be much more highly expressed for whatever reason, whether it was induced by diet or environment, or it's something that's more genetic, that we can have precise treatment for that particular group. And so I think the shortest answer to your question is multiple factors are involved. We acknowledge that race is not something that is purely genetic, but it is an important or sort of categorization that helps us to understand some of the differences that we're seeing.
Thanks. There are two questions. One from Kathy Ann Kowalski. Based on the anterior-posterior difference, should doctors take biopsies both front and back? And a related question from Anitra Brown, does it make a difference in treatment or survival rate if the tumor is indeed located in the front or the back of the prostate?
Right. So the first part of that is, yeah, I would absolutely say yes. So not very many people have studied this. So I think it's absolutely worth it to look in different cohorts to see how consistent this difference in tumor location is. And that could absolutely inform how we move about in terms of the standards for biopsies. So I know the standards have changed before. There was a time where I think there were less sampling from one particular direction, and having more information tells us, hey, it might be worth it to also go, from the other side. So yes, I do think that is something that needs to be reviewed in the urology and oncology world. The second part of that question related to treatment response. So based on that particular study, I'm not aware that the response specifically is different, but they did highlight that there were certain molecular differences or that were different based on the location. So it is possible that if a patient is diagnosed with an anterior tumor, for instance, that maybe his treatment approach may need to be different, right? So he's less likely to have AR expression, for instance, so anti-AR treatments just won't work. But I don't have definitive evidence that the response is different.
Right. And I have one final question. Based on what you've observed with cytokines, neutrophils, and macrophages in the context of prostate cancer in Black versus white populations, are any of these characteristics, can they be carried over to any other type of cancers in terms of these sorts of racial disparities?
Absolutely, yes. So, multiple cancers have looked at these types of, markers. Macrophages in particular have been very studied in the M2 world. So a lot of people are typically familiar with what M2 macrophages are doing. They're helping the cancer, they're dampening the immune response that might help to kill the cancer, et cetera. But M1 macrophages, for instance, are not as well studied in any of the cancer types. And so perhaps some of the future work we'll do in understanding what role it might have in prostate cancer can then be also carried over to other cancer subtypes.
Great. Well, thank you very much, Janielle, for a really interesting talk. Really appreciate it.